Have you ever stopped to think about how the entire instruction manual for you, a 2-meter-long strand of DNA, gets crammed inside a single microscopic cell? Well, today, we're diving into one of biology's most incredible packaging miracles. So let's start with a problem that is frankly mind-boggling. If you were to take the DNA from just one of your cells and stretch it out, it would be about as tall as you are. And yet all of that has to fit inside the cell's nucleus, a space so tiny you can't even see it. It's an insane puzzle that every single one of your cells has to solve. And the answer? It lies in this absolutely remarkable substance called chromatin. You find it in complex organisms like us. And here's the key. It isn't just pure DNA. It's a super dynamic complex of DNA and a whole bunch of proteins, all working together to package our genetic code with an efficiency that's almost hard to believe. Okay, so how in the world does this work? Here's how we're going to break it down. First, we're going to really get a feel for the sheer scale of this problem. Then we'll dive into the solution, zooming in layer by layer, from that very first fold of DNA all the way down to the tiny chemical switches that are running the whole show. All right, first up, let's really appreciate this engineering challenge. I mean, packing DNA isn't like just cramming dirty laundry into a suitcase. The cell has to be able to find and read specific genes at a moment's notice. So this system has to be both unbelievably compact and perfectly accessible. And the secret to this incredible feat isn't actually the DNA itself. Turns out, your nucleus contains an equal amount of helper proteins by mass, and most of these are a special type called histones. You could say they're the real MVPs of DNA packaging. So how do they work? Well, just think of histones as these tiny microscopic spools that have a positive charge. And your DNA, it has a negative charge. And as we all know, opposites attract. It's this beautiful, simple principle of physics that's the foundation for winding up that 2-meter thread of life. Okay, so the cell has these protein spools. The next obvious question is, how do they actually organize the DNA? This is where we get to the first fold, called the nucleosome, and it's really the fundamental building block of all chromatin. You know, when scientists first got a peek at chromatin under an electron microscope, they saw something that was just elegant. The whole structure looked exactly like beads on a string. It was a simple description, but that one observation kickstarted our entire understanding of this whole process. So each one of those beads is a structure we call a nucleosome. At its heart, you've got a core of 8 histone proteins, and the DNA wraps around it about 1.5 times, kind of like thread on a spool. Then a little bit of unwound DNA, the linker, connects it to the next bead down the line. And the efficiency here is just wild. This very first step, just wrapping the DNA around these histone spools, it shortens the DNA's length by almost 7 times. That is an incredible first level of compression. But of course, a 7-fold reduction is nowhere near enough to get the job done. So let's level up and see how this string of beads then coils on itself to get even more compact. The whole beads on a string structure gets twisted and coiled up into a much thicker, denser fiber. It measures about 30 nanometers across, and this represents the second major level of organization for our DNA. Now, for a long time, scientists actually debated how this fiber was built. The early idea was the solenoid model, that it was a simple, hollow coil, kind of like an old telephone cord. But more recent evidence points to a more complex zigzag model where 2 stacks of nucleosomes actually twist around each other, forming a sort of second, much thicker double helix. And what's really brilliant here is that the structure isn't just random. The cell uses different structures for different jobs. Active DNA that's currently being read tends to form that more flexible zigzag structure. But inactive, silent DNA, it uses the simpler, more compact solenoid structure. The packaging itself is actually a form of information. So this brings us to a really critical question. If the DNA is packed this tightly, how in the world does the cell get in there to read a specific gene when it needs to? Think about it like this. Imagine your DNA is a massive library, but every single book is shrink-wrapped shut and jammed onto the shelf. How do you open just the one book you need at exactly the right time without messing up all the others? Well, the cell's solution is just beautifully elegant. It temporarily removes the packaging. It creates these little clearings called nucleosome-free zones, which exposes the important parts of the DNA, like the on switch for a gene, and makes it accessible to the cell's machinery. And the process is like a targeted demolition job. First, a signal comes in. That signal recruits a team of specialized proteins, these chromatin remodeling complexes, that basically act like a molecular cleanup crew. They physically slide the nucleosomes out of the way, clearing a path so the gene-reading machinery can get in and do its job. Okay, let's zoom in one last time to the most intricate level of control. This is where the cell uses tiny chemical switches to tell that remodeling machinery exactly what to do and when to do it. The secret is actually in the histones themselves. Sticking out from each one of these nucleosome cores are these long, flexible tails. And they're not just random floppy bits. They're basically a sophisticated control panel for the whole operation. The cell can decorate these tails with a whole variety of different chemical tags. These marks create a complex language, often called the histone code, that other proteins floating around in the cell can then read and interpret. So, for example, one of the most common tags is something called an acetyl group. When the cell adds these tags, a process called acetylation, it neutralizes some of that positive charge on the histone tails. This makes them loosen their grip on the DNA, the chromatin gets less compact, and it basically acts like a green light for turning a gene on. And as you might guess, the opposite is also true. Removing those acetyl tags, deacetylation, lets the tails clamp back down tightly on the DNA. This stabilizes that compact structure, and it acts as a red light, effectively silencing the gene and shutting it down. And all of this leaves us with a pretty fascinating thought. Our DNA sequence, that's our blueprint, right? But this histone code, it's like the foreman on the job site deciding which parts of the blueprint actually get used. So if we could learn to edit this code, to add or remove these tiny chemical marks, we might be able to control which genes are on or off. And that could completely change the future of how we treat disease.